Big six markers, they are often based on the required practicals in sciences, and one of those in Biology Paper 1 is the factors affecting the rate of photosynthesis. This is everything you need to know about that practical. Obviously, let's start with the purpose. The reason we do this practical is to investigate how a factor affects the rate of photosynthesis. Normally, the factor that we change is light intensity. And normally the plant that we use is an aquatic plant, something like pondweed. The reason we use an aquatic plant is because as it photosynthesizes and produces oxygen, we can then see the oxygen produced in bubbles and count those bubbles to give us an estimate of the rate of photosynthesis. Let's talk about the control variables. So if we're changing light intensity on purpose, then everything else that could affect the rate of photosynthesis needs to be kept the same. That includes temperature, the concentration of carbon dioxide, and the amount of chlorophyll available. Obviously, using the same plant every time, we're going to have the same amount of chlorophyll. We submerge our pondweed not in water, but in something called sodium hydrogen carbonate instead. Sodium hydrogen carbonate, like it says at the end of the name, carbonate, contains a buttload of carbon dioxide. That provides all the fuel that we need for the photosynthesis. We then take our light source and place it next to our pondweed at a set distance, something like 10 centimeters, measured with a ruler. We always allow a certain amount of time, something like 2 to 5 minutes, for the pondweed to acclimatise, to adjust its rate of photosynthesis to the current light intensity. We then count the number of bubbles produced by the pondweed in a set amount of time, something like 30 seconds. The more bubbles the pondweed produces, the more photosynthesis that's occurring, because those bubbles would be bubbles of oxygen, one of the products of photosynthesis. Of course, you'd expect the most bubbles to be produced when the light source is very close to the pondweed, when there's lots of light intensity, lots of light available for photosynthesis. Then we take the light source and move it further away, 20 centimeters. Repeat again and repeat again at every set distance up to, say, a meter. You remember earlier I said we need to make sure we control the temperature? Well, to do that, ensure that the light bulb we use is not a filament bulb which gives off a lot of heat. Ensure instead you use an LED lamp, because an LED lamp doesn't give off any heat. It will not affect the temperature. Of course, with any practical, to improve your results, repeat it and calculate a mean for each distance. And that's that really. If you've got any topic requests, pop them in the comments. If not, follow Mr. Wells, because I'll be posting a video at least once or twice a day for every single day until the end of the science exams. Then we repeat those steps.